This is the first of a series of several videos that are going to review basic biochemistry from what you learned in biology in your 9th grade year, such that you are ready for biomedical science. I'm attempting to do this in pretty short order and give you the basics so that you can have these in your mind as we begin our more advanced study of anatomy and especially of physiology. Recalling that the basic structure of life, the cell, is a very complex structure made up almost entirely of water and then these organic biomolecules. It's necessary to have some familiarity with these basic building blocks of biochemicals in order to proceed. Please remember from your biology class that we categorized these biomolecules into 4 major groups: carbohydrates, lipids, proteins, and nucleic acids. Biochemistry is the study of these chemical interactions that occur in living cells, and biochemists spend a tremendous amount of time studying the structure and physical properties of these 4 classes of biomolecules. The reason for that is because we need to understand how the cell operates, but it's also possible to use this information to manufacture new drugs by taking these similar features or features that are similar to those we find in nature and incorporating those into the design of new kinds of treatments. Fortunately, biomolecules that are of interest to us are made up almost entirely of very few elements. Those are carbon, hydrogen, nitrogen, oxygen, and to a lesser extent phosphorus and sulfur. You can remember this by the acronym CHNOPS. One feature of these elements is that they all have a tendency to bond with one another covalently. And so if you look at their location on the periodic table, here, 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 and then right down in here, you see this very small area. These are reasonably small atoms. The overwhelming variety of these biomolecules are covalently bonded, and in the presence of an aqueous medium, and enzymes are very easily rearranged. So given the complexity of even very simple life forms, it's important to recognize that millions of chemical reactions are constantly occurring and are necessary for the elaborate pathways that have evolved over millions of years. Metabolism, defined as the sum of all these reactions, take place between almost entirely these organic molecules. These organic molecules have some common characteristics. Organic molecules are almost always associated with living things. They always contain carbon, and specifically, they contain carbon-carbon bonds and carbon-hydrogen bonds. And so that is kind of the hallmark that we use for identifying organic molecules. These are frequently large molecules with many carbon and carbon-hydrogen bonds, and oxygen to a lesser extent. And they almost always have covalent bonds, so meaning that they share electrons, and that this attraction of each of the atoms for the shared electrons is what keeps those molecules together. Reviewing for you that macro means large, and when we're talking about monomers, we're talking about the building blocks of these larger biochemical classes. When we divide them into the 4 categories, carbohydrates, lipids, proteins, and nucleic acids, we can break each of these categories down into looking at the monomers, looking at the building blocks, and then see how they interact as they as they join one to another to another. In order to talk about that, I want to review the concept of anabolism. Anabolism is the term that we use for the process of building macromolecules. Although these molecules are very complex, the reactions that join them together and break them apart are very simple. Anabolic reactions link together monomers into long chains via a kind of reaction that we call a dehydration reaction, sometimes called a dehydration synthesis. The requirements for this reaction is that one reactant is going to lose a hydrogen, and that's shown right here, and the other reactant is going to lose an OH. And I don't know that it's especially important that you recognize that it could be this OH and this hydrogen, or vice versa. The result of that is that water's formed, and we form a covalent bond that links together what were two monomers into one monomer. So the water, the hydrogen and the OH, is lost, and the oxygen between them covalently bonds these two monomers together into, in this case, a dimer. This case, it happens to be a dimer of sucrose. These reactions are enzyme catalyzed, of course, and can take place many hundreds of times to link many, many polymers into a very long single polymer or into multiple polymers that will interact together. All of the biochemical classes that we're going to study join their monomers together in this way. So this is a very important concept of this dehydration synthesis. Catabolism is the process of breaking down macromolecules. In this way, we can take these big molecules and turn them into their smaller integral parts. This catabolic reaction is the reverse, basically, of what it was involved in anabolism. In this case, water is added, not as a molecule, but broken down into the hydrogen and the OH group. And in that case, the hydrogen adds to one of the monomers and the OH to the other. So in this case, we have two molecules being broken down from one. This specific type of reaction is called hydrolysis, and you may remember that hydro means water and lysis means to break, and so in this case water is broken down into a hydrogen and an OH, and in the process it frees that, in this case, dimer into two individual molecules. Again, this requires an enzyme in order to happen, and so what the enzyme is going to do is it's going to lower the activation energy, make it easier for this reaction to take place, easier for molecules to achieve the energy, the activation energy required to break apart. The first of the macromolecule classes that we're going to talk about are carbohydrates, and carbohydrates are extremely important because they are sugars and groups of sugars. And the purpose of carbohydrates is energy storage and to provide energy. Your body uses carbohydrates as fuel. The most direct, simple way to give your body a fuel that your cells will use easily is to give it carbohydrates. It includes three major types: monosaccharides, which are simple sugars, sometimes called single sugars, sources of very quick energy. Your body can metabolize these very quickly. Disaccharides are double sugars. They're sugars where we have two monomers stuck together, two monosaccharides stuck together via dehydration. And they tend to be useful for short-term storage, but they're also a means by which the body will frequently transport sugars from place to place. Or in this case, it could be a plant. It probably would be more likely to be a plant, actually. And then we finally have polysaccharides, where we have many, many monomers, many simple sugars that are joined together. And this is usually a form of long-term storage and a form of structural fiber that frequently is important to both plants and animals. To take it from the first, monosaccharides are the most common, monosaccharides are glucose, fructose, and galactose. These are all classified as hexoses, meaning they have 6 carbons. If you remember, the chemical formula for glucose is C6H12O6. That's also the molecular formula for fructose and galactose. Although they are organized, their structures are different. They have the same chemical formula. You may remember that we called that structural isomerism. Same formula, different arrangement. These monosaccharides, again, are quick energy. They're the easiest form of fuel to digest. They exist as cyclic structures. We're not going to go into why that is true, but if you take biochemistry, you'll learn why that happens, why that structure becomes the primary route by which these molecules take their shape. Some other monosaccharides worth noting are ribose and deoxyribose. These are pentoses, meaning they have 5 carbons instead of 6. And those are important because they are the building blocks of RNA and DNA. Disaccharides. The most common disaccharides are maltose, lactose, and sucrose. Maltose is made up of 2 molecules of glucose bonded together in a dehydration reaction. Sucrose is a glucose and a fructose, and lactose is a glucose and a galactose. Worth noting is that maltose and sucrose are made by plants, as is glucose, for the most part. And lactose is a disaccharide that's made by animals, specifically mammals, as a means of providing energy through milk to the young. Again, I said earlier that disaccharides are a means by which plants frequently will transport sugars from the site where they're made, which would usually be a leaf, occasionally a photosynthetic stem, but usually a leaf, to some place where it's going to have long-term storage. This is, again, 2 monosaccharides joined by dehydration synthesis, and they are held in that position for transport, and when they reach the site at which they're going to be used, the plant, or in this case perhaps the animal, will produce an enzyme necessary to break them apart. Polysaccharides are part of a very large subgroup of carbohydrates. Glucose polysaccharides are the ones that we're going to be studying, and some examples of them include glycogen, where we have long chains of glucoses joined together. Glycogen is produced by animals. You have glycogen that's stored around your liver and muscle, and if you're a male, you have glycogen stored in sperm as a source of energy. Cellulose is strictly a plant polysaccharide. Huge numbers of glucoses polymerized into a long straight chain that are responsible for primarily the makeup of the cell wall. Humans, if you remember, are not able to digest cellulose. It serves in our diet as fiber, but it is the most abundant organic molecule. So all these plants that you see, each one of their cells is surrounded by a cell wall made of cellulose, and sometimes another polysaccharide relative that is lignin, responsible to give extra special strength to cell walls. Chitin is another kind of polysaccharide. It's a glucose polymer, but it is slightly different in the way it's joined together and serves as an exoskeleton for crustaceans and insects. The shells on shrimp, the shells on lobster have a lot of chitin associated with them, again, as well as hard-shelled insects. Generally, this is indigestible by humans, but we can modify this chitin and turn it into a molecule that's taken by lots of people, me included, called glucosamine, and serves as I won't say a drug, but it is a nutritional supplement that promotes the well-being and the formation of cartilage. We can talk about that more when we get to the skeletal system. To take a look at the organization of polysaccharides in plants, if you were to look here at a cross-section of a leaf and see these plant cells in an array, if we were to look closely at just the plant cell wall, we would see these overlapping cellulose fibers. And if we were to take a look at one of these individual fibers up close, we'd see that there were a tremendous number of what are called microfibrils, which are simply long strands of glucose that are joined together over and over and over and over again in these individual polymers made up of many, many glucose monomers. I think that when we talk about cellulose, it's perhaps up to 5,000 of them in a single one of these strands that in turn is going to wind around other strands to make up the actual cellulose fiber. If we were to look in a plant cell or in an animal cell, if we were to look at another kind of plant cell, in this case it would be a potato, we would see the cell wall as we saw before, but we could also, perhaps in the case of a potato, see these granules of starch that are made up of two different kinds of polysaccharide fibers. You would see not only the unbranched, arranged in a slightly different way than we see in cellulose, and we would then see some that are branched and kind of held together in a sort of meshwork. So again, you have these these glucose polysaccharides, both branched and unbranched, and if you were to look at them, if you were to make an emulsion and take a look at them under the microscope, you could use iodine and you would actually see them. They would actually stain kind of a darkish color when they pick up the iodine. You have this identical kind of granule in not only potatoes, but we also have that stored in our liver. The second class of macromolecules that I want to talk about in this video is lipids. Lipids are insoluble in water, and that's probably their most overriding feature. You can remember from the past that you can kind of remember how solubility works in that like dissolves like. So things that are polar are soluble in other things that are polar. In this case, lipids are nonpolar, and they are soluble in nonpolar solvents as well. There are four major kinds of lipids that we're going to talk about briefly. Triglycerides are important because that is an energy storage molecule that we use or that animals use and plants as well. Triglycerides can be classified as fats and oils. We'll talk about that a little bit. They also serve as insulation. Triglycerides are made up of a molecule of glycerol, which is actually not a lipid. It's an alcohol molecule, but joined to that alcohol molecule we have three fatty acids. Another kind of Lipid that we're going to talk about is a phospholipid, and phospholipids are important because it is phospholipids that make up the cell membrane, the major structure that controls what gets into a cell from what gets out of a cell. Very important feature of cells. Lipids are also classified into steroids, which makes them important cell signaling molecules, primarily as hormones, not only sex hormones, but other kinds of hormones. Many are classified as steroids or as lipid-based hormones. And finally, waxes. And waxes don't tend to be as important to us in biomed, but they're certainly important from the standpoint of plants, because having a waxy coating on the leaf of a plant prevents it from being associated or having a lot of water loss. It allows it to contain and hold its water so it doesn't wilt. It's able to maintain plenty of water in the central vacuole. And of course, you're familiar with the production of wax by bees to provide a structure in their hives in which they are going to hatch and raise their young. Occasionally we talk about waxes associated with furry animals and humans, and if you watch CSI, you might occasionally hear about a body becoming waxy because of the way it's been stored rather than buried, but that's kind of an unsavory way of getting waxes. In order to take a look at triglycerides, what they look like, you can see here that we have two different kinds of fatty acids associated or shown here. We have unsaturated fatty acids. Fatty acids are very, very long, and if you note, they have a huge number of carbon-carbon-carbon hydrogen bonds. And if you remember when we talked about organic molecules, we said that they all have carbon-carbon-carbon hydrogen bonds. The reason that fats, particularly fatty acids, are so good at long-term energy storage is because there is a tremendous amount of potential energy held in these bonds. This is classified as unsaturated because it doesn't have the full amount of hydrogen it could have because of the presence of these double bonds. And so unsaturated fatty acids have one or more double bonds. One would be unsaturated. Multiple would be polyunsaturated. So this would be a polyunsaturated fatty acid. And saturated fatty acid is down here. Again, a very excellent source of energy storage, and in this case we have all carbon-carbon single bonds. And so it's saturated because it has the maximum amount of hydrogen it could possibly have. For this reason, when we put these molecules together, when we bind them to this glycerol, you can see that the unsaturated fatty acids have little kinks where these double bonds are located, which means that they have a lower density. They are more liquid, and so frequently when we look at oils, we can classify them as being high in polyunsaturates because they are liquids at room temperature, whereas lots of saturated fatty acids, because there are no kinks in their carbon-carbon chains, are able to pack more tightly and exist at room temperature as solids. Very quickly, phospholipids occur by having a phosphate group associated with a derivative of glycerol, and these long fatty acids associated with that. And in this case, sometimes they have double bonds, sometimes they don't. But this idea of having a polar end, and if you remember, the more oxygen you have, you set up more situations for hydrogen bonding to occur. So in this case, the fact that we've replaced our glycerol or one of the fatty acids with a phosphate group means that we have a polar head and these long nonpolar tails that arrange themselves in a cell in such a way that the nonpolar tails point toward each other. Again, like dissolves like, or like is attracted to like. And since the outside of a cell is usually watery in nature and the inside of a cell, I've got this switched around, is watery in nature, these molecules line up like little soldiers with their polar ends near where the water would be located and their non-polar ends directed away. So the formation of a bilayer to produce this plasma membrane or this cell membrane exists primarily as a very, very, very micro thin layer of lipid that regulates what's going to enter and what's going to not enter cells. Steroids are derived from cholesterol, and here you can see cholesterol has a characteristic shape that consists of four carbon rings that are attached together. So you can limit your cholesterol in your diet. And when you do that, your liver will make some cholesterol. Some people have difficulty with the buildup of cholesterol, and we'll talk about that a little bit more later on in the year. But cholesterol, for our purposes, is going to be a necessary precursor for many of the hormones that are useful for us, specifically the sex hormones, but not exclusively sex hormones. These are important for allowing an organism to communicate, cellular communication within the organism. And it's important to note that one of the ways by which these sex hormones are able to get into and out of cell is by simply diffusing through the cell membrane because of their lipid nature. The last thing I want to talk about here are waxes. You do make a waxy kind of substance in your ear produced by some glands in your ear called ceruminous glands. The difference between fats and waxes is that waxes have extremely long lipid chains, carbon-carbon-carbon-hydrogen chains, more than 20 in length. And so when we said that I suppose short chains or highly polyunsaturated chains are liquids, straight chains, or fully saturated chains are solids. The hard solids consist of very, very long carbon-carbon chains. This is as far as we're going to go with our first video. There will be a second video talking about proteins, enzymes, and nucleic acids. I hope this has been a useful refresher for you, and I will be happy to discuss any of this in class.